resume the recording. So what we're going to do today is we'll finish up the last lecture on ecophysiology, and then I'm going to give a brief description of our first paper summary assignment, because as you hopefully remember, it's coming right up. It's due a week from Wednesday. So I'll go over that. You guys can ask questions, and then we'll start the soil habitat lecture after that. So I will go ahead and share screen. So we left off on this slide talking about resistant structures. So as you remember on the previous slide, it talked about some different ways of scavenging nutrients as a one response for microbes to avoid poor conditions or to help compensate for poor conditions. Okay, so the alternative is to just avoid them altogether, right? And so many of you, I hope, are aware that there's a range of different kinds of resistance structures. Sort of the classical one that's taught in general microbiology courses are endospores, right? And so gram-positive groups like Bacillus, Clostridium, things like that will form these very hard resistance structures. But this has actually evolved multiple times in the microbial lineages. So you can also see cysts that are formed. There's in cyanobacteria, there's aconites. So, oops, excuse me, something's going on here. Sorry about that. I try to use my mouse to advance things. Sometimes things go a little crazy. All right, so what these all have in common is that they're typically more tolerant of conditions than regularly growing cells, which we refer to as vegetative cells, okay? So there's actual physical transformations either in part of the microorganism or the entire cell changes and becomes this resistant structure. Typically, these are non-metabolic cells, so that's the big change. Vegetative, is indicating that they're growing, and active, whereas these are going to be non-metabolically active cells. They're basically just sitting there waiting for conditions to get right. Okay. All right. This is actually under very elaborate regulatory genetic control. So this has been studied best in Bacillus, looking at endospore formation. And so as you might imagine, the genes that have been identified for this are called SPO genes for sporulation. And there's over 30 different gene loci that are involved in this that are known. And so this just shows sort of a picture from a classical textbook on this. You don't need to memorize the details of this, but just essentially what you need to know is that to convert from a vegetative cell in this state all the way to the dormancy stage here at this end on the right-hand side requires many different steps and is initiated by many different genetic genes being turned on and regulation. Is there a question?
Yeah, I was just wondering, what does global regulatory control mean?
So what I refer to there is that these cells are undergoing genetic, transcriptional, translational changes. And this is very radical. As you can see, the entire cell changes shape, it changes size. And so this is not just a single gene turning on. This is actually a suite of dozens of genes that are being turned on or turned off. And that requires quite a lot of genetic regulation. If you remember from your genetics course or your introduction, cell biology course, right? So you've got to have transcriptional regulators that are turning these on and off, and it all has to be orchestrated in synchrony. And so that's what I mean in terms of global regulatory control. You don't have to know the details of it for this course. This isn't a genetics course, but it's important for you to understand the complexity of it. Does that make sense?
Thank you for clarifying. Yes,
Sure.
definitely.
And it's great. Thank you, Johan, for asking your question. Because if anybody has questions during the course of my lecture, don't be shy. I was speaking with a student earlier who was saying that they didn't want to ask questions because they feel like they would look foolish. And I know sometimes I'll say things that may assume certain knowledge that you guys have. And when I can't see you in person, I can't tell if there's confused looks on people's faces. So please do raise your hand or speak up to ask questions. Because if you have a question, it's quite likely that somebody else has the same question. So I appreciate that question. Another strategy that has evolved is motility, right? So microorganisms, especially bacteria and archaea, are very small, right? And so typically to get their nutrients, they rely on diffusion across their cell wall and cell membrane of nutrients and carbon and all sorts of things. What that means is that they often run out locally of those nutrients. So there's kind of a sphere of influence around a cell. And if the nutrients are depleted in that zone just immediately outside the cell, then they're out of luck, right? They would have to stop growing. But if they could move to a new location where the nutrients are not depleted, via motility, this can allow them to explore new areas and find new nutrients. The diffusion is very slow and leads to this local limitation. Okay, so being able to move around is important, and there's multiple ways to move. Some of them are actually in response to chemicals, or in the case of photosynthetic organisms, sometimes it's light, that the cells have certain receptors which allow them to cue in on light levels or chemical availability and regulate their motility that way. This requires them to sense their environment using what we would consider to be quite simple systems, but these have been working for millions to billions of years. So obviously they do the trick for these microbes. Most often, these are two-component regulatory systems. And I'm going to go over this in a little more detail in a couple of slides. But the important thing to know is that there's the two components. The first is a sensor, and the second is the regulator, or the thing that causes a change to occur inside the cell. Sensors are typically kinase proteins. Who remembers what a kinase does? You should have learned this in cell biology. What does a kinase enzyme do?
Does it remove phosphorus group?
So it actually removes it in some cases, but really it is adding it in most cases. So the kinase adds the phosphorus, and then another enzyme, the phosphatase, removes it. Okay.
Yeah.
So the sensor kinase, however, it is obtaining the phosphorus by removing it from high energy molecules that have phosphorus. And does anybody remember what those are? It's something that often has 2 or even 3 phosphoruses attached to it, and it's our energy currency inside cells.
ATP?
Exactly. So a kinase will often grab one of those high energy phosphates off of the ATP, converting that to ADP, and then it sticks the ATP's phosphate group onto a new molecule. And what that allows is for a conformational change, which can then lead to another step of regulation. So this is what that looks like. So these are membrane spanning proteins, just like a cartoon here. But in response to some sort of environmental signal, you might have a binding that occurs, right, in a binding pocket here. this causes a change, a conformational change, a structural change in that protein. And in the presence of ATP, these sensor kinases can then phosphorylate the response regulator protein. That phosphorylation will then make it an active transcriptional regulator. And so this is just another very simple schematic, but as you remember, right, RNA polymerase is often under very strict control, and you can have in the operon where you have genes being spread around, and where the genes are located, and the upstream transcriptional regulatory elements like the promoter region and the operator region. This is where that response regulator will bind, and it'll either upregulate or downregulate whatever gene is in question here. And that will either turn on RNA polymerase's activity or suppress it. Okay. And then ultimately you need to get the phosphate back off so that you can regenerate your response regulator. And that's the phosphatase that I mentioned a minute ago. Okay. So these two component regulatory systems are very common. They've evolved long ago. And so there's lots of different examples. So for example, in chemotaxis and phototaxis that I just alluded to, sporulation that I just described is under two component regulatory control. Quorum sensing, which is a form of bacterial communication that we'll talk more about later this semester. Biofilm formation, lots of nutrient uptake systems, stress responses, antibiotic resistance, more complicated interactions with other types of organisms can be regulated by these. So there's just a whole suite of sort of day-to-day activity where the bacteria, in this case, let's just say for bacteria, are sensing their environment and responded using these two component systems. And there was a fairly recent paper that found over 240 genes in a genomic database that had homology to the two component regulatory genes. So it turns out that you can see similar gene sequences and identify these two components, the sensor kinase and the response regulatory, in genomic databases. And they found hundreds of these. So these likely evolved millions and millions of years ago and play a huge role in microbes responding to their environment. All right, so that's the end of that lecture. Are there any questions about that? All right. So let me end that. All right. All right. Sorry, Angel, I didn't see your chat. When I'm lecturing, it's hard to see, but I see that there. So thank you for contributing earlier. All right, let's go back. Now I'll share screen. Okay, so before we start the rest of today's lecture, I wanted to just go over next week's paper summary assignment. So the first thing is you guys need to download and read the papers. Okay, so the first paper we're reading next week, I think, is this Oxenreiter paper. Okay, so the syllabus has detailed instructions on how to download it from the OneSearch library website for Cal State Long Beach. If you have any questions or you cannot access the papers, let me know and I can help you, or you can contact the research librarians. They can help you. So you're going to download this paper and you're going to read it. I don't expect you to understand every word in the papers, although this first paper I feel is pretty accessible to you guys. So do your best in reading it. You don't have to be an expert on it. But the idea is that we're going to read these papers. you're going to summarize them so that you demonstrate to me that you can kind of understand the paper well enough to synthesize it and come up with a short summary. And then we'll critique them a bit, and then we're going to discuss it in class. So about 3/4 of one page. So this is a 1-page summary, single spaced. If you write about 3/4 of the page, you would be doing the summary. So that's the majority of it, is the summary. What I want you to identify are the questions or the hypothesis or the main objective or topics of the paper. Okay, so those need to be clearly stated. You're going to want to articulate the main methods. So you don't have to describe in great detail every single method they use. What I'm interested in you doing is summarizing, saying things like they used 16S ribosomal RNA sequencing, dilution culturing, and fluorescence microscopy to analyze samples from X. Okay, something like that would be a succinct summary, but it clearly states that you know what methods were used in the paper. Okay. And you're going to talk about like what those key elements, those key factors are. And probably the most important thing you can do for primary research articles, which is mostly what we're reading for our summaries this semester, is identifying the results. And where do we go to identify or see the main results for any primary literature article? Does anybody know? What should you look at?
The figures and the interpretation of the figures that's unbiased.
Yes, so the figures or tables, right? That's where the data are, right? And so you're going to want to read those, look them over, and you don't necessarily take their word for what it means. You should think about what that seems to be showing to you before you even read their description of the results. So what I actually like to do when I read a manuscript for the first time is to kind of just give it a quick look through and look at the figures and the tables and see if I can figure out what the major findings are without even reading their abstract or their introduction or methods and results. Now I understand, reading this can be challenging, and so you may have to read the paper more than once. So you're definitely not going to want to wait until the night before to write these summaries. Okay, so give yourself plenty of time. At least you should be doing it by this weekend, I would say. Okay. And then you want to have what you see as the conclusions, or what the authors described as the key findings or conclusions to be made from it. Okay, so 3/4 of the page roughly is going to be spent summarizing all of these points. And then the last bit will be kind of a critique. This is the harder part of the assignment, I understand. When you're first reading papers, it's really hard to know whether you should believe what's being presented or whether the science is good, so to speak. You might have questions. And this is really what I want you to do is spend this last bit, last maybe quarter of the paper, thinking about the paper, writing questions, and you can talk about, well, I was unclear in this section. They could have done a better job doing that, right? So do the authors clearly and concisely make their points? Give an example, okay? Another key thing, and this is probably the most challenging thing I'm asking you to do with this assignment, is identifying an assumption. So scientists are human beings, and all science builds on previous science. And so there will always be underlying assumptions in any study. We're not ever really in a position to be starting from scratch with no preconceived notions or assumptions. When we do science as researchers, we have to figure out how to ask a question and then what the best methods to use to answer that question would be. And there's often underlying assumptions in the choices that are made in how to investigate the question, what methods, and then underlying assumptions potentially in terms of what the results might mean. So what I'd like you to do is take a stab at finding at least one assumption made by the authors during their study. Okay. Then you're going to want to just talk about the writing. Was it easy to read and straightforward? Or maybe it was really hard, dense, hard to read. How could they improve things? And do the figures or tables that show the data, are they clear? Is it obvious? Are they legible and easy to see, like all the different lines or bars or whatever? These are the kinds of things I want you to think about. And then I want you to ask, what's the best thing about the paper and what was the worst thing about reading the paper? So you, in this quarter of a page, you want to like go through each and every one of these, and basically one sentence is sufficient for each of these. Okay. And then the last thing you want to do is ask a question. So what question occurred to you while you were reading it? It could be, I didn't understand how that method works. How does that method work? Or it could be a scientific question, which is kind of what I'm hoping to get you to over the course of the semester, where you actually ask a question about maybe the findings, like the authors say this. I'm wondering about the next step, or I'm wondering what that would mean in this regard, right? So ideally, you'd be able to ask a question as if the author was sitting in the room right in front of you. And you could say, hey, I really like this part of your paper. What about X, Y, or Z? So those are the key things to do in this last portion of the summary, okay? And you have to have a question, okay? So you can look at the grading rubric and see what each of these elements are worth in the assignment. It's posted on Beachboard. But the other thing that's important about reading the paper is that you get 5 points for participating in the in-class discussion next Wednesday. And if all else fails, the easiest thing for you to do to participate either verbally or in the chat is to just ask your question. Right? If you've already written this down, you can just articulate it in class and boom, you get your 5 points. Okay? So next Wednesday, we'll be discussing this paper, and I'm going to invite as many of you as are comfortable or able to turn on your cameras. We're not going to have a PowerPoint. We're just going to talk about the paper. We might share screens once or twice to look at the figures or something like that. But basically, we're going to have a discussion. If we were in person, we'd be circling up the chairs in the room and we'd just be talking about things. My goal is to not do all the talking. My goal is for you guys to ask questions and potentially even answer each other's questions or bring up points. And then I will guide you through the discussion of the paper as we go along. Are there any questions about the assignment for that? So you'll upload the summaries to Beachboard. I believe I set the deadline as 10 a.m. the morning before class on Wednesday, a week from Wednesday. Okay. And so that way I know you did the reading in advance and you're able to do that. Okay. So this will be good in its own right, but it will also prepare you to be reading and thinking critically about scientific articles and preparing you for the term paper you'll be writing at the end of the semester. Okay.
I have a quick question.
Yes, please.
Is the summary going to be a single spaced summary?
So the 1-page limit is for single spaced. If
Okay,
you want to
perfect.
double space it, it can be longer then. But yeah.
Well, thank you.
But it's, you know, you're typing it, so you can go ahead and make it single spaced. Yep. Other questions? Okay, so as things go along, if you you're reading the paper, you're having a really hard time with it, come to office hours next Monday morning or shoot me an email if you have questions and are having difficulty with getting going. But don't feel like you have to understand the paper fully. Okay, we're just learning how to do this. Okay. All right. All right. So if there aren't any other questions, we're going to move on and talk about microbial habitats. So we'll be talking about these for the next few lectures. We'll start with kind of a brief overview of all habitats, and then most of today will be on soils and sediments, so sort of terrestrial habitats. Next time we'll talk about aquatic habitats, both freshwater and marine, and then we'll talk about extreme habitats next Monday. So habitats, of course, have a huge impact on microorganisms. And when we study microorganisms in the environment, one of the first questions that you should ask is, are these indigenous or local organisms, or did they come from somewhere else? So we use these terms allochthonous and autochthonous. So if these organisms that are found, let's say in a soil, have been there for generations, then we would call them locals or autochthonous. But let's say we're studying a river. Many of the organisms in a water sample from a river might have been washed down from way upstream, and that would be referred to as an allochthonous microorganism. So these are terms that are important for us to consider, and also factors that are yeah, so Jenny, you asked, are you going to upload this PowerPoint slide? I did. Was it not visible for you guys?
No, I couldn't see it either.
I wonder if it, hang on a second. I wonder if it, it does that thing where it makes it, oh, sorry about that, where it may not make it visible right away. Oh my, it didn't upload it at all. Okay, well, that's not good. I uploaded it yesterday. That's so bizarre. Okay, well, let's just take a 2-second break and I will post the video, so you should be able to go back and fill things in. All right, so one environment we don't talk about a whole lot is the air. So how good an environment do you think, or a habitat do you envision the air would be? Or let me rephrase it, because it says there it's traditionally not thought hospitable. What might be some of the boundaries or barriers to air being a good habitat for microorganisms?
There's no medium for them to attach to?
Okay. That's not necessarily 100% true, because there may be some particles that get dispersed, like dust gets blown around, and you will often see microbes attached to those particles. But you're right, the air in and of itself is not. And interestingly enough, it's thought that you may have heard of like cloud seeding. In trying to get rain to form. Oftentimes rain droplets do form on dust particles which have microbes attached to them. So it can play an important role in the world's weather. However, the majority of microbes are not like hanging out in the air most of the time. And if they are there, they're often there very transiently or just short time, right? So for example, I hope you're all aware, like if you've ever like stepped on a puffball fungus, or maybe if you were in microlab and you opened your plate and there was fungus all over it and little poofs of clouds of, the spores came into the air. Usually your instructor will tell you, put the lid back on, don't open your plates, because it'll contaminate the whole lab with fungal spores. And the reason for that is they disperse very well. And so those resistant structures are often very common. Now, this was the view for a long time, but then there was a study done, and that's this study here done, gosh, I guess it's been about 10 years. I can't remember what year this was, but And what they did is they went to the top of Mount Bachelor, which is kind of like Mount Shasta, right, or Mount Lassen. It's one of the big volcanoes up in the Cascades in the Sierra Nevada chain. And so they went up to the top of Mount Bachelor and they sampled the air there. And there you're at, 10,000 feet or whatever. So you're way up there. And what they were able to determine, combining sampling from there as well as looking at models of weather and dispersal, is they were able to show that both bacteria and archaea get moved all the way from Asia over to North America on a fairly frequent basis. And so sometimes it goes up here over the poles. And of course, this is a fairly narrow gap, the Bering Strait, although that's kind of skewed because of the map, the two-dimensional map here. But it can even be extended, all the way across, thousands and thousands of miles of ocean. So the wind and air is a habitat in some senses, but the reality is most of the organisms will not be active while they're there. They're just passing through and they might be moving to a new habitat. Next week, we're going to talk about, or next time, we're going to talk about those aquatic habitats, both freshwater and salty marine. And then today, we're really focusing on terrestrial habitats, soils and sediments, and then we'll finish off. We may not get there today, but we'll talk about hard rock subsurface environments as well. But we'll start with some very superficial, literally superficial rock environments. And these are so-called lithic habitats. So lithos is rock. And so what you can see here is that microbes can actually exist on the surface of rocks. Who knows what this is a picture in the upper right-hand corner of?
Lichen?
Lichen. And thank you, Jocelyn. And what does anybody know about lichen? What are lichens? Anybody know anything?
A symbiotic relationship between fungus and bacteria.
So they're actually, they can be bacteria, but they're usually a phototroph. So that can be either cyanobacteria or it could be algae. But it's a fungus and some sort of phototroph, yes. So typically rocks are not very hospitable environments. There's not really a lot of nutrients unless you can leach some of those nutrients out. And so having a photosynthetic organism like these lichens, or what you can see here, these dark patches on the surface of this desert, these are crusts of cyanobacteria that form biofilms or mats on the surface of the ground or rocks. So having that photosynthetic component allows them to survive in a low carbon environment. What else do you think characterizes like living on this rock might be? What other aspects of that habitat would they encounter aside from not having much like ground to get stuff from?
Maybe like water retention.
Okay, because so they're likely to not have much water at all, right? If you're on this rock, unless it just rained, and incidentally, when it does rain, lichens will often swell up and absorb a bunch of moisture, but eventually they're going to dry out again, right? And so, yeah, so water retention is a big deal. What else do they need to worry about if they're photosynthetic?
Being able to get sunlight?
They need sunlight, but what's the downside of sort of sun?
It could dry things out.
Okay. What other aspects of the sun's rays do we need to
Is
worry
it-
about?
Is it the UV that damages the bacteria?
Yeah, and so you can actually see some of these pigments. Some of these pigments are actually UV photoprotective pigments. So these guys are going to be under desiccation stress, low water. And that's going to create osmotic stress, right? So all the nutrients and sugars and things inside will want to go, you know, will want to rush out if you've got any moisture around them, and all the moisture will want to get sucked right out when it's really dry. But high ultraviolet radiation is a big deal. The dark coloration here is actually caused in many cases by a pigment. And this is the pigment that I studied for my dissertation work, produced by mat forming cyanobacteria. So another really kind of boutique environment that microbes inhabit is so-called crypto-endolithic communities. These live inside rocks, but we're talking about the surface. Later today or next time, we're going to talk about deep subsurface environments. But right now, what we're talking about are communities that live just under the surface of a rock. And based on the color of this, what kind of organisms do you think we're looking at here?
Probably cyano, some kind of photosynthetic organism.
Yeah. So in this case, we often have fungi, and they form like these black bands. And then you'll have some sort of algae. So it may be cyanobacteria or it may be eukaryotic algae that live there. This is a gypsum crust from a salt pond that I do research in down in, well, I used to once upon a time, down in Guerrero Negro, which is about halfway down the Baja Peninsula. And you can see the pink color here is caused by halophilic microorganisms, and we'll touch on those in a couple lectures. But inside this gypsum crust, there's a cyanobacterial community. This is actually an Antarctic crust. So there's dry, I know we normally think of Antarctica as being covered with ice, but there's dry air portion of Antarctica as well. In fact, it's called the Dry Valleys region. And these are what we would refer to as cold deserts.